this opportunity to share our work with you. So I work for ACASO, which is the acronym actually stands for the Goddess of Healing, and it stands for the Austere Environments Consortium for the Enhanced Sepsis Outcome, which is a mouthful. But so what we do is we study sepsis. So sepsis is a life-threatening condition that when you have an infection, your body, instead of just fighting the infection, can start attacking itself and the healthy tissue in your body. And you can have organ dysfunction from it, think liver, kidneys, and you can also die from it. So 20% of all global deaths are associated with sepsis. And this, it really affects areas of low to middle income countries where 85% of all sepsis cases are in those regions. And so there are a lot of deaths that occur in those low regions, and it's mainly because it's an austere environment. So the resources do not fit the need. They don't have the antibiotic treatments. But also, a lot of the research that is done for sepsis is done in developed countries, using subjects from developed countries. And so you don't get that genetic variability. And also, they get sick from infections that are seen in developed countries. So a lot of people have tried to take the research that's done and bring it into these regions, and the results don't translate. It doesn't work. And so what we've done at ACASO, ACASO was started in 2010, we've created this clinical network where a lot of our sites, studying sepsis especially, are in low to middle income countries. So those are all the ones with the cross that is red. And so we've built this system where we run observational studies, and we collect data on subjects that come in with sepsis, with the goal of developing point of care devices. So right now, the current paradigm is that if somebody comes into the hospital, they're sick, you isolate, you take a blood sample from them, isolate cells, do a blood culture, see if bacteria will grow, and then use that to see what types of antibiotics will work on killing off that bacteria. This process takes 4 to 5 days. But right now, this is the only type of information that the clinicians have in order to be able to really define what that person is sick with. What we're trying to do is instead take a point of care device, take a drop of blood, and instead of focusing on this whole defining what the pathogen is, what we're looking is at the host response, and how can we take that host response to define what type of treatment they should be getting. So we run an observational study in all of our different sites, and we collect blood samples over time. And we freeze those samples, have them shipped back to Maryland, and we analyze them for genes, proteins, and metabolites. Each one of them is giving a unique interpretation of that host response over time as the person is sick. So I was asked to give a little bit of a comparison between what happens in academia versus industry. And so what I've tried to do here is show you just basically, so at ACASO, we really focus, and all of our work is in terms of projects. So we usually start off with defining our project and take it all the way down, if it gets to this stage down here, to point of care assay development. So what we really need to do in order to create, just basically to keep that project running, are these gray modules. But what we try to do in each and every instance is to build in this discovery phase, where we're taking the data, looking at it through using mathematical techniques, and then trying to relate that to biological interpretation. This is the most this is the part where it's most like a graduate student PhD. But for us, we have this whole other things, and it's really trying to balance that, how much are we putting in just to getting our project finished, to how much can we build in research-wise. And it's that research component that actually gives you your ideas for the next projects that are coming up. So it keeps the projects going. Another difference between industry and math in academia is it's not just you working on your PhD project. We have a whole host of scientists, all with different backgrounds. These are all different people that we work with within ACASO, all with different backgrounds, and we work together as a team to get these projects finished. And communication is huge. Being able to discuss your ideas and discuss your results, and being able to tell that to scientists and computational people, and also support. What kind of support do you need in order to keep these projects going? Communication is critical. Another thing that it took me a little bit of time to figure out, but you need to advocate for your ideas. In order to get your ideas incorporated into any kind of work structure, they need to come into this where you're defining the project. So really advocating your ideas, and it can't be like you have an idea and you say, oh, well, I just think it'll be really cool to try this out. You have to come with a either scientific background or some kind of financial justification for why the company should be interested in doing these ideas. Another difference comes in with when you're in the translation stage. So let's say you found this really cool discovery mathematically, you were able to translate it biologically, and now we want to work to create a point-of-care device for it. You basically have to dwindle it down to about 8 to 10 biomarkers, come up with a predictive model for it that will work on your point-of-care assay device. And so for point-of-care assay development, working with people who actually do the experiments on the devices, what they really like working with are evolutionary decision trees. Well, decision trees in general. And the reason is because at each node here, you have just one biomarker, and you're interested in whether or not it's above or below a threshold. And so when you're designing an assay around that, they just have to be accurate and precise around that one cut. Instead of if you're using a continuous model where you have to be accurate and precise along a large swath of quantities for it. And I've really enjoyed working with evolutionary decision trees for this part, which build genetic algorithms into discovering the trees, and it's been a lot of fun. Another difference is that it's not just one project, but multiple projects all at the same time and at different stages. So when I first started, I was put onto 3 different projects, and we met every 2 weeks, which meant I was doing presentations, 3 presentations every 2 weeks. So as I got a result, it went straight into a PowerPoint presentation. So optimizing time management and organizational skills are huge. So during COVID, it was a really, really interesting time. And if you look, these right here are all of our COVID sites, and the number of sites actually doubled. And so we were just, really focused on getting everything out the door. So we were mainly focused on getting this stage done and this stage done, and very little discovery was going on. And after a while, it gets, you're doing the same things over and over again it gets pretty boring. And I felt like my creativity was stifled. There was nothing really that I could do besides keep those trains going. So I had a serious talk with the director and told her, I was like, I can keep these going, but if I can get like 1 or 2 people under me that I can supervise on projects, for this mathematical discovery and biological interpretation stage, that would be wonderful. And so she agreed. And so what I was able to do, I reached out to Richard Bertram, who's head of the biomath department, and to see if there were any students who would be interested. And so we were able to bring in 3 different students. We brought in Mehran, Francis, and Nicole. So Mehran, when he graduated, we actually hired him. Francis is finishing up his PhD right now. And Nicole, when she graduated, she went on to a postdoc. At the same exact time, we hired a biostatistician and a bioinformaticist. And this formed the Biomath Research and Development Group, which I lead. And it's been a lot of fun. So some of the math that we're doing here, I'll talk a little bit about two applications. One is clustering, and then the other one is going to be dynamical systems. But for clustering, so sepsis is really treated as a one-size-fits-all. Basically, think of cancer, 70 years ago. If you got cancer, it was a one-size-fits-all treatment. Nowadays, it's by what is infected, what subtype it is. If it's breast cancer, you can see the commercials for, HR positive, HER2 negative, and there's a precision medicine that's associated with each of those types. And sepsis is not there yet. But there is a lot of heterogeneity in the patient population. And a lot of people doing research will kind of use that as a cop-out. Well, they'll say, oh, it's just because of heterogeneity that our results didn't work. So what we try to do is cluster these subjects, and can we find those different classes of subjects for precision medicine? Another motivation for clustering is with these, we do a lot with RNA samples. Each RNA sample can compute 30,000 genes. You can get quantifications for that many genes, which makes it really hard to work with. And so what we can do is then cluster these genes based on correlation. And so the method that I like for clustering is spectral clustering. And the reason is because it's graph-based. So here you can see this graph of nodes. And what you do with spectral clustering is create a similarity matrix. So define some kind of similarity between nodes. It can be either correlation-based, it could be based on distance, and then use that for your graph Laplacian and calculate the eigenvalues. And spectral clustering has a really nice property that says the largest gap that you see within the eigenvalues is going to predict how many clusters you have. So this is going to be 4 clusters, and you can see those 4 clusters within the data set that's there. So we've expanded on this idea and done 2 different projects. One is multi-layer bundling, where instead of just looking at, so this is a correlation-based similarity, and instead of just looking at that one type of clustering, what if you took all of the top eigengaps that you have and layer that information into your graph? So here you can see that these nodes are ones that have survived the first several layers of clustering, that they still remain within the same grouping. And this allows us, this is for our gene network, and we can actually take it and use it at the different layers and build that into our analysis. Another method is multi-scalar clustering, and this is a distance-based similarity. So distance has a property where it's inversely related to the similarity. So it needs to go through a function where you take that distance and then map low distances to high similarity. Now the interesting part comes in actually from this sigma, which is just describing the width of that kernel. And what you can see the effect that it has on just the definition of what is similar, but then also on the eigenvalue curves that you have. So here I've plotted them out for all of the different sigmas. And with it, you can see the number of clusters changes as you traverse sigma. And you can look at these persistent regions and be able to map them out as to what their clusterings are like as you go from like the local scale all the way to a global scale within the data. Another thing that we're interested in is dynamical systems. And this came about from different results that we were seeing within our data, where you could see subjects that were going from a state of critical illness up to health. And then even within our proteins, this is one of the first plots I did just to data clean the proteins, but I thought it was really beautiful because you can see at time 0, the points are all spread out everywhere. And over time, the healthy people who survive, they all come to this steady state. And so a friend of mine that I work with, he had found this paper, and it was all about disease space mapping. And he was so excited about it. And he's like, look, it can trace people going from a state of comfort to sick all the way through recovery. And I'm like, this is a phase plane diagram with a trajectory on it. And they were showing it as a cartoon. And I was like, they're interested in doing this? I'll go in and show them all the stuff that I have. So I brought in the Strogat's book on nonlinear dynamics to them, and I was showing it to them. I was so excited and everything. And it was met with a hard no. I had never been told no before from them, but they're like, this seems way too complicated. It's going to be way too hard to develop, and we don't have somebody with the time to put towards it and everything. And so when I got one of the students, I made sure one of their projects was with an ODE system. So, and this is Francis's project. And so what he was doing was he took a model that Nick Kogan and Angela Jarrett had worked on, describing the different attributes within the immune system with respect to bacteria. And he expanded it so that it contained our biomarkers. So we were able to plug in our biomarkers that we were capturing into the system. And then as we find clusters of subjects, we can look to see how the different components change within them. And so he should be defending this research in a few weeks, so I'm gonna leave it at that. And I'll show you one more project that we have going on with dynamical systems. And it had to do with this work that was done here. So this work was actually started by somebody in my lab, Josh Chenoweth, and Carlo Colantoni from Johns Hopkins. And there's always the problem of dimensionality reduction with respect to genes. And so they did this non-negative matrix factorization on the genes. And what it does, this program creates 30 patterns within your genes. So think of these as vectors. And what they were able to see was that when you map pattern 4 versus pattern 23, it really traced out this really nice trajectory of moving from a critical state to a state of health. And so this is work that Miran was doing, and he was seeing, okay, well, we have these two patterns. Can we model them phenomenologically? So it's a very simple system, a lot of Volterra ODE model. You have your two variables, 4 parameters, and we played the game. We saw, what were the best fits for these trajectories. But what I think was really cool is what we decided to do after that. So not only were we looking at the best fits, but actually, as you traverse those parameter spaces, how does the cost function change? What is the distance between your actual model and the data that you have? How is that going to change as you traverse that space? And here you can define that as this dynamical system cost fitting landscape. So what it's doing is telling you information about that trajectory through the lens of the Lotko-Volterra ODE model. And what you can do then is calculate this cost-fitting landscape for each of the models. And as they differ, those cost fits are going to differ. And you're able to capture key information about those shapes and trajectories. And so then you can take that, use that as data, right? And then here we've taken each of those cost-fitting landscapes. We did a UMAP of it, and you can see the majority of people who survive are in this upper corner. We can apply our clustering algorithms to it. So here you can see cluster 1 are maybe those people who survive. This is the actual really messy, dirty data. And this is the data prediction. And each of these, what was really nice was what it did was it broke down those people who died into about 5 different groups, all with different types of projections that are going through that space. And so it allowed us to cluster those subjects of death into just being able to pick up some of that biology. And what's also really neat, so if you plot it just for the initial condition, take the first point that you have for it, that blue group where everyone survives, you can see that it overlaps tremendously with all the different groups. You're not able to capture that separation unless you're looking through the lens of those trajectories. And so that was just to give you a taste of some of the work that we're working on. There's a lot more, but I'll leave it at that. But I did want to show a little bit about how I got to a CAISO. So, because I think, especially students, I'm sure you guys go to a lot of talks and you see the beautiful work that everyone's doing, and you don't hear the backstory of how they got to where they are. And so I wanted to share a little bit of that. So I did my PhD work here at FSU with Monica Hurdall, looking at the folding patterns of the brain. On the mathematical side, I was using Turing reaction diffusion systems, which relate pattern formation in terms of the size and shape of a domain. And so the domain I was interested in was a prolate spheroid and prolate spheroidal surface. And what I was able to do is take those results and relate that back to the evolutionary development of the brain. Because as the brain developed, there's an internal ventricle that elongates, and so I could capture that eccentricity in terms of one parameter of the prolate spheroid. And the initial folds that form actually fall out from that analysis. So it was really, really cool. It was a fun experience. At the time, I was very terrified just because of being a PhD student, but it was a really fun experience. It was the first time I could really see the interaction with the math and the biology. After I graduated, I had the two-body problem. My husband is a chemist, and we ended up moving to Maryland for his job. And when I got there, you think greater DC area, tons of jobs and everything. 2 weeks after I got there was a hiring freeze. And so basically no job options whatsoever. And so I was at home for a little while. And then I came across, so it was actually Richard Bertram sent me a job announcement for a position at NIH. And so I interviewed there and got the position, and it's a 5-year postdoctoral fellow. It's fellowship, which is unusual, but it's really, really nice to develop new skills. It is a government agency, so that you don't do any grant writing, for better or for worse, because you also don't get practice with grant writing. But he basically let us choose whatever we wanted to work on, as long as it was data-driven and had some dynamical aspects to it. So I was working in graph theory and the alignment of, in looking at pancreatic islet cells and how the structure of them changes with diabetes and also during development. And to tell you the truth, I worked my butt off at this postdoc because I basically did not want that same thing to happen where I couldn't find a job afterwards. And so as I was publishing, I was going to meetings, and towards the end, there was an offer for a PI position. And so I went and interviewed. It was a two-day interview process. It was very nerve-wracking. I got through all of it. Everything was going wonderfully. And a month before I was to leave FSU, a month before I was to leave NIH, the position fell through. The funding disappeared, and they weren't going to hire anybody. So I was back at ground zero with nothing. And so I tried some other things and applied for some other things in research, and nothing was happening. And I said, you know what, I don't need this. I loved teaching while I was here at FSU. I'll go into teaching. I have no problems with that. And so then it was really interesting because I ran into one of the parents at the daycare that I was at. This is Subbu Krishnan. And I told him what I was going through, and he's like, no, you need to go in and talk to my director, who's Danielle Clark. She was just telling me yesterday, we need a mathematician, which that you never hear, ever. And he's like, you have to go in and talk to her. I'm gonna send you the job description and everything, and you talk to her. I saw the job description, I was like RNA-seq and stuff, and I'm like, no, I'm done, And I went in and spoke with her. I showed her the stuff that I've done in the past and everything. And she's like, this would be perfect. What we need is someone not to, just run algorithms. what sepsis needs is somebody who can develop algorithms to drive the science forward and everything. And so she's just been wonderful to work with. I always like to say why I am at CASO. This is always a gut check for me all the time. And I love challenging problems with interesting constraints. I love learning new science all the time. And for me, what's really important is empowering others, helping others. I love doing that. And what also helps is the ACASO mission. It's something tangible, something you can see the results that are happening from the work that you're doing. So with that, I'd like to say thank you. This is the ACASO group of everybody that I work with. So thank you.
Thanks, Debbie. Are there any questions? We have a couple minutes for questions.
I have a question regarding your financial systems graphs, especially if you could go back to the one where it has the cycle of going into sickness and back to recovery.
Oh, this one right here?
Yeah. So I'm kind of curious, like on the y-axis is health. Is that based on like a qualitative or quantitative?
Yeah, so this was a cartoon that was, so that's the, if you want to look at the actual article, that's the citation for it. But yeah, they were looking at pathogen load and this made up version of health, what it was. It was a really interesting article where, you know, they were looking in mice in their infection, and they were able to quantitate a little bit with health, but for the main thing that they were trying to get through was this idea, this caricature of being able to traverse, you know, a system, which is just reinventing. For me, it's reinventing the wheel. It's already been, yeah. So, but it is in PLOS biology. So, yeah.
Cool. Thank you.
So you have gained a lot of experience for the past 10 years or so. Do you have any advice you want to share with our graduate students?
So, advice for graduate students. I mean, a lot of it is hard work getting published, going to meetings, really networking. You never know who you're going to meet at a meeting. Another thing is when you're looking for a job, tell everybody. For me, it was a parent at the daycare, right? But you never know. It could be your kooky uncle whose next door neighbor owns a company. You never know. But it's really, another thing is, especially, so coming from a math background, a lot of people don't understand what we're able to do. And I found that a lot. They would always ask me, oh, so you're a biostatistician? I'm like, no, I'm not. And so really learning, it's like almost the language of how to get through it. What I would also recommend is taking, even if you can, like a course in data science, just so you have some credential that says data science, because sometimes that can get you into the door just for that conversation. And then you can show them, look, I can also do all this other stuff. But that would be my advice. But so that was the thing why I wanted to get students from here. I knew what, you know, the caliber was. I knew that they were mathematically rigorous and had computational skills. And so, I mean, for me, it was a gold mine. So yeah.
All right, well, on that note, let's thank Debbie again.